Hey guys, so today we're going to go ahead and go over chapter 12, and that's basically DNA. We're going to go over chapter 12.1, 2, and 3, so you're going to get all of that information here. What I suggest that you do is take out your final exam study guide, and as I'm going through the lecture, write down the answers to the final exam study guide, okay? And so that's what I'm going to actually have expected for tomorrow, okay, is that chapter 12 final exam study guide portion done. All right, so we'll go ahead and start ourselves off here. Okay, so like I said before, this chapter is going to be about DNA. We talked about this in our review game, but in the middle of the 1900s, scientists were asking questions about genes. They didn't actually know what they were made from, where they came from. They knew of the macromolecules in our bodies, but they didn't actually know what was responsible for heredity. At the time, they mostly believed that proteins had to be the molecules that made it out because there are like 32 different amino acids that make up the different proteins in your body. Whereas DNA, you only repeat 4 basic subunits. So DNA seems too monotonous and simple for that to make up all the differences in humans and anything living. So we had some different scientists that went ahead and researched this. So our first one we'll talk about is Griffith, and basically he looked at the pneumonia virus, and he figured out that there were 2 different strains of the virus. There was this smooth strain, and when injected into mice, the mice would die, and then there was this rough strain. When that was injected into mice, they would live. Oops, there we go. If he heated that lethal smooth strain, we actually found that the mouse survived. So the heat-killed bacteria were no longer lethal, okay? They allowed the mice to live. After that, he went ahead and he took the dead smooth strain and he combined it with the live rough strain. And when he mixed those together, he found that the mice died, which was really interesting. He's like, why is this happening? When he looked inside the mouse, he then found that the live lethal strain was there, even though he killed it with the heat. So somehow that lethal strain was passed into the live strain. Griffith called this process of one strain being passed into another one transformation, because the bacteria had been changed permanently into another one. So transformation, think of transformers. You're transferring one thing into another one. But he didn't know the factor. So Griffith is responsible for transformation, but what was being transformed? Was it proteins, lipids, carbohydrates, nucleic acids? He didn't know. So after that, more scientists are looking at this experiment and saying, okay, I wonder. So let's try to repeat these experiments, but let's do something different. So we know that there's transformation happening, but now let's go ahead and add a digestive enzyme to our different macromolecules. So Avery used digestive enzymes on polysaccharides, lipids, and RNA, and when he did that, he found that transformation still occurred. The mice. However, when he treated that same sample and he put enzymes that would destroy DNA, he found that the mice lived. So transformation wasn't happening. And DNA was the molecule that caused the genetic change, not a protein, not a carbohydrate, and not a lipid. So Griffith's experiment showed that genetic material could be passed between bacteria and cause change, okay? So Griffith showed transformation. Avery's experiment showed, and he's the one with the digestive enzymes, that genetic material was DNA. So scientists are skeptical people, and that's a scientific attitude that we have to have. We can't just trust 2 guys' experiments. We have to continue to experiment in order to believe that they exist. So our next people, 1952, Hershey and Chase experimented with a virus that infected bacteria, and as you know, that's a bacteriophage. And we knew that bacteriophages were made out of proteins, and they're made out of DNA. So proteins is what people used to think was the cause of heredity. So what they did is they put genetic markers on the proteins, or they just put markers on the proteins. Well, they put the sulfur on the protein, okay, and so that sulfur just stuck to the protein coat, and then they put the phosphorus marker on the DNA. So they basically thought that if DNA was the cause of transformation, we're going to find that phosphorus inside the bacteriophage. But if it's the proteins causing the transformation. We're going to find proteins inside of that bacteriophage, or the sulfur marker. Well, in the end, they actually found that the phosphorus was found inside of the new bacteria, and so DNA had to have been the cause of transformation. So it showed only DNA, not the protein entering the cell during infection. So a conclusion of Hershey and Chase's experiment is that genetic material in the virus was DNA and not proteins. So now we can talk about a little bit about the role of DNA. So the DNA that makes up genes must be capable of storing, copying, and transmitting genetic information in the cell. So it can't just be there and then not used. We have to store that information safely. We have to be able to copy that information. Then we have to be able to read that information and make proteins from it. So, role of DNA. The genetic material stores information needed in every living cell. So even though your body is made up of tons of different of cells, they all have the same exact copy of DNA. So my heart cell, even though it performs a specific function, it still has the instructions to maybe perform the skeletal cell instructions. Okay, but that's just not read in the heart. So before a cell can divide, its genetic information has to be copied, right? So in order for us to go through mitosis, we have to go through that S phase. So remember, that's the synthesis phase, the making of more DNA. We have to copy it so our new cells have the exact same copy of DNA. So each daughter cell has to receive a complete copy of genetic information. Okay, so now we're going to go ahead and address the structure of DNA and what DNA looks like, who founded DNA, and everything like that. After that, we'll talk about replicating DNA. So we said that one of the rules of DNA is making sure that we can replicate it for mitosis and meiosis. So we'll go ahead and get into chapter 12.2 and 12.3. All right, so this is pretty much what DNA looks like. DNA, what you should think of, is a double helix molecule, okay? So that means that it's double-stranded, and then the helix is basically that twisted shape. So we have a couple people that are responsible for this. The X-ray experiments by Rosalind Franklin, that's that woman on the left, led to James Watson and Francis Crick to the discovery of the actual structure of DNA. So Rosalind Franklin took the pictures of the DNA, whereas Watson and Crick actually kind of figured out the structure of it. What's really sad about Rosalind Franklin's life is that she died before the Nobel Prize was given out for finding the structure of DNA, and so Watson and Crick were the only people that actually received the Nobel Prize. Very cool people, very smart people. And it's just unfortunate that she wasn't able to live to see them get that prize for that. Anyhow, so DNA, or nucleic acids, are made out of a subunit called nucleotides, and that's something you guys learned in chapter 2. In nucleotides, we have a phosphate group, a sugar, and a nitrogen base. Specifically in DNA, the sugar is going to be deoxyribose sugar. We have the nitrogen bases. We have 4 different nitrogen bases. We have an A, which stands for adenine. We have G, which stands for guanine, C for cytosine, and T for thymine. Okay, again, we say that deoxyribonucleic acid, as you can see here, there are 2 sides of it, so we consider it double-stranded. So the backbone, or the sides of the ladder, are going to be made up of phosphates and sugars, okay? So all that purple stuff there. Then we have Chargaff's rule. So Chargaff's rule, he's this guy that basically saw that A is always going to equal T, so adenine is always paired with thymine, guanine is always paired with cytosine. At that time, no one knew why. Now we know it's because, like I just said, adenine always pairs, or the amount of A's equals the amount of T's. Amount of G's always equals the amount of C's. And we didn't know why then, but we know now that it's because adenine will always pair with thymine, and guanine will always pair with cytosine. Okay, so basically, if I told you that we had 30% of our DNA was made up of adenine, you would identify that, or you would exactly know that 30% had to be made up of thymine. And if we add those 2 together, that's 60% of our DNA. Well, if 60% of our DNA is made up of the A's and the T's, that means that 40% must be made up of G's and C's, and we would see that at 20% and 20%. So again, we call it a double helix molecule because it's all twisted up, and that's just because of the bonds that form between them. So hydrogen bonds actually allow the adenine to pair with the thymine and guanine to pair with the cytosine, and that's what keeps that double strand, or those two strands, together. Okay, so now we're going to go ahead and continue more about the DNA and chromosomes and the way they look. So a DNA molecule in bacteria is going to be single-stranded, and it's going to be circular and found in the cytoplasm. Remember, bacteria and prokaryotes, they don't have a nucleus. Okay, DNA in eukaryotes, it's much more complex. So if the diameter of DNA is 2 nanometers, was wide as a fishing line, it might stretch as far as 21.2 kilometers in length, which would all be packed into a nucleus, the equivalent size of 25 centimeters in diameter. So we have so many different base pairs, and you can imagine that that has to be packaged really tightly in order to fit into the nucleus of the cell. So think about it. If you could just take a piece of string, how would you fit it into that little egg container? You'd have to crumble it up and wrinkle it up, or kind of just, yeah, basically crumble it up so that it would all fit. So that's basically what happens to DNA. DNA is in multiple chromosome bundles, and it's found in the nucleus. So we kind of wrap it up really tightly in our chromosomes so that it can be basically divided later. Eukaryotic chromosomes are made of DNA and proteins called histones. Together, the DNA and histone proteins form a bead-like structure called the nucleosomes. And so you can kind of see that there, the DNA, then being wrapped around those purple histones, the proteins, then get wrapped up really tightly together, and that's going to form our chromosomes. Okay, nucleosomes, like I said, packed together, and then we just see this picture continue on to form the chromosomes and our chromatin. So in the dividing cells, we're going to see chromosomes. In the non-dividing cells, we're going to see chromatin. So how is DNA copied? The structure of DNA explains how it can be copied. Each strand has all the info needed to construct the matching other half. So if the strands are separated, base pairing rules allow you to fill the complementary bases. So we have to kind of separate it and then add the different bases to it. So sites where strands separate and replication occur are called replication forks. So they're the forks that basically, so here's this right here, that's the replication fork. That's basically where it separates that DNA strand. So we can see here's one strand of the DNA. And then this is DNA polymerase. DNA polymerase is basically what adds the complementary end. And you see another one right here. So they work in opposite directions. So the first thing that happens in replication, the enzyme has to unzip. So the DNA polymerase has to unzip the strand of DNA. So we have to break those hydrogen bonds. 2, the DNA polymerase then joins nucleotides using the original strand as a template and spell checks for errors. So basically it just adds nucleotides to one side of the strand while the other DNA polymerase will work on the other strand. Copying happens in opposite directions along the two strands and in multiple places at once. So imagine if you just had 2 DNA polymerases working in opposite ends. With our DNA being so long, that would be almost ridiculous to think about. So they work in opposite directions multiple times because we have so much DNA. So replication, when we think of replicating DNA, you need to think of DNA polymerase. That ACE always stands for an enzyme. DNA tells you what it's making, because we will eventually talk about RNA polymerase where RNA is being made. So replication happens with DNA polymerase. And guys, that is it for this chapter. Remember, I expect that your chapter 12 final exam study guide questions should be done for tomorrow. Okay, and you should have been able to get that by looking or watching this video. I hope you have a nice night, and if you have any questions, just come in early and ask me.